Hello and welcome back to a Coach Carter education series on skeletal muscle structure and function. Today we're going to tackle video number 2 on sliding filament theory. Now this is our mechanistic understanding of how skeletal muscle contracts, how it produces force, and how it causes movement. Now, this theory was originally established in the mid-1950s and really hasn't changed a whole lot since then, which is pretty cool. We've added a lot of understanding since then, but we haven't really found any glaring misconceptions. So first, we're going to anchor back to our sarcomere from video number 1. Now remember, this is the smallest contractile unit of skeletal muscle. You have a bunch of these sarcomeres linked in series to form myofibrils, which are arranged in parallel to form the cord of the muscle fiber. Now remember, the muscle fiber is the cellular level of skeletal muscle. Even though the sarcomere is smaller, that can be confusing sometimes. The contractile myofilaments are actin and myosin that make up a sarcomere, form visual bands, discs, and zones that are just going to help us a little bit later to understand the structural basis of contraction. So within the structure of a sarcomere, we have actin and myosin, or thin and thick filaments. You might hear them called sometimes. Now, these are very crude drawings to help us just understand how they're arranged. That'll help us then understand actual sliding filament theory. Now, each sarcomere contains multiple of these thick myosin filaments that are arranged in a bunch of hexagons connected together. Every single one of our thick filaments, our myosin filaments, are surrounded by 6 thin actin filaments. And these thin actin filaments can not only interact with the one myosin, but actually adjacent myosin as well, which allows us to be really efficient. And in other animals and other muscle types, like smooth muscle, the ratio actually changes depending on the purpose of that muscle. Now, if we were to look at an actual computer simulation of what an arrangement of this actin and myosin looks like, it's something a little more like this. So, a lot going on in there, but all I want you to think about is we have our thick filament, okay, surrounded by these thin filaments that are going to pull on each other to form contractions. Now, before the muscle can actually contract, we have to get a signal from somewhere. And this signal arrives in the form of an action potential from a nerve. We're going to go into this a little more in video 3, but for now, just know we have this action potential from a nerve that then is able to go through this network. Now, if we trace the network, we have an action potential that arrives at what's called the sarcolemma, which is our specialized cell membrane for muscle fiber cells. It then goes through this network of transverse tubules or T-tubules that can reach all regions of the fiber. This is really important because it allows the fiber to contract all at the same time, so we don't have this kind of misfiring altogether. As the action potential reaches certain areas, we have voltage-sensitive receptors in the tubules that open up these calcium release channels in our sarcoplasmic reticulum. You might have heard of an endoplasmic reticulum when going over basic cell parts, cell organelles. Our sarcoplasmic reticulum is our specialized endoplasmic reticulum in a muscle fiber. So just think, we have an action potential that comes from a nerve. We're going to go over how that works in video 3, but it goes through the sarcolemma, through our transverse tubules or T-tubules, and activates voltage-sensitive receptors in the T-tubules to open up calcium release channels in our sarcoplasmic reticulum. So we have this flood, or this rapidly released amount of calcium ions into our sarcoplasm. Once we have this flood of calcium ions into our sarcoplasm, which is our specialized cytoplasm, our cytosol of our skeletal muscles, we can come up here to step number 1. Okay, so calcium ions are present. So the position of our tropomyosin exposes these actin-myosin binding sites. So tropomyosin is a binding protein that surrounds our actin filaments, and when calcium is present, there's a conformational change in the tropomyosin to expose binding sites. Now, our myosin's already ready to go. It actively binds to the actin to form cross bridges. Okay, now this process itself doesn't actually require any. The myosin is already ready to go. It's in this state where it'll just actively bind once the site is open. The myosin heads then, after they're attached, will bend. This does consume energy, releasing ADP and an inorganic phosphate, and the actin is pulled inward. Okay, we'll talk about what that means in a second. Then we have another ATP that will come and bind to the myosin head, and the myosin head is then released from the actin. Okay, this ATP is then used to recock the myosin head for the next contraction. Okay, so back to number 1. We have this flood of calcium ions. Okay, the calcium ions cause a conformational change in tropomyosin to expose binding sites on the actin. From there, myosin, boom, like a magnet, attaches to the actin, okay, and it then cocks the head, causing a pulling, and it releases ADP and an inorganic phosphate. Then a new ATP binds and releases the myosin head, which is then spent, and the myosin head is recocked. Okay, so I want you to imagine it like this. Over on the right side, we have our myosin filament in red. Okay, looking at the top here. Now, our myosin filament is going to stretch out in 2 directions, and it's going to have actin coming in on both sides. Okay, so what's going to happen is, as this power stroke happens, or when the myosin head bends, it's going to pull actin filaments from both sides towards its midline, or the M line. Okay, this causes a shortening. There's not actually a squeezing of any of the filaments. They're simply sliding past each other because the myosin is pulling on the actin. Now, the actin is anchored to the Z lines on either end of our sarcomere, and it pulls these Z lines closer together, which is a contraction at a very, very microscopic scale. Now, just imagine this happening thousands, tens of thousands, hundreds of thousands, even millions of times, all at the same time. Causes our muscle to fully contract. Okay, so if we look at the top, this is when it's relaxed. We have our myosin filaments and our actin filaments out to the side. As it contracts, our actin filaments get pulled toward the midline. Okay, the Z lines come closer together, and we have a contraction. Okay, what you can think about is the H zone here is the space where there is only myosin showing. There's no overlapping. Okay, it's only myosin showing. And you'll notice the H zone shortens with contraction because the actin is being pulled toward our midline. Okay, our A band just refers to the actual full myosin thick filament. And this does not change, right? Because there's no squeezing going on. It's just sliding back and forth during the contraction. Our I band refers to the space where there's only actin. There's no overlap of actin and myosin. Now, this will also shorten similar to our H zone because the actin gets pulled over the myosin. Okay, so let's go again. We have flood of calcium. the calcium causes conformational change in tropomyosin. The myosin binds to the actin. The myosin then pulls, power stroke, okay? That's the contraction piece. The myosin head then releases and reloads. Attach, power stroke, release, reload, okay? All right, video 2. Sarcomere, smallest contractile unit of muscle, right? This is going to be a test question somewhere. Just commit that to memory. The sarcomere is divided into these different lines, zones, and discs that are just visual markers to help us understand contractile physiology. Every sarcomere contains multiple thick myosin filaments arranged in a hexagonal lattice surrounded by 6 thin actin filaments. Now remember, these actin filaments extend across and interact with multiple myosin filaments. Okay, so they share. Once an actin potential reaches a muscle fiber from the nerve, which will be video 3, we travel down the sarcolemma into the T-tubules, okay, that results in our sarcoplasmic reticulum releasing calcium ions into the cell. And that's where sliding filament theory goes. Okay? It describes how the myosin heads pull the actin filaments toward the center, the M line. Just remember, midline. Therefore, shortening the sarcomeres, resulting in muscle contraction. Okay? That was a lot. Feel free to go through it again if you need to. Video 3.5 is going to be a full review, okay, after we go over the nerve and muscle interaction in video 3. But I hope you enjoyed this one, and I'll see you guys in the next one.